Okay, now I will start a little bit with the QTL before we have a break. Is that okay?
Yeah.
So when we talk about the comparison of monogenic and polygenic inheritance, just to give you an idea, when you have one gene and two alleles, we have a very simple situation of two markers here and one capital A, small a heterozygous in the middle. In the F2, without environmental variation, you have the three classes, as you can see, and there's also here a peak, but not very clearly. Even with some variation, environmental variation, you still can distinguish the three genotypes from the situation that you have one gene, two markers. If you have two genes, heterozygous, you see that the situation is a lot more complicated. In the F2, we have 5 classes, and supplemented with the effect of the environment, you can see you have a sort of continuous variation. So here it is getting very difficult, and when you have, in this case, you have 5 markers, then you can see that in the F2, plus the environmental effect, it is impossible to distinguish the different genotypes. In that case, it means we talk about QTLs, quantitative trait loci. Now, to understand a little bit these QTLs in a genetical network, we have to realize that one gene can correspond to one QTL, or one QTL has 3 genes, 1, 2, and 3, that control together a single QTL. In the case of gene 3, you see that not only QTL-A, but also QTL-C is involved. In other words, you can see multiple genes are involved to make one QTL, but also different QTLs can control the expression or the product of one gene. In other words, you end up with a regulatory network which is a lot more complicated when you have quantitative trait loci. And years ago, when I taught my students about these kind of things, there's very nice research about 50 years ago in yeast. We're not talking about yeast, but yeast, everything is possible because there's so much more known about the genes, genome of yeast, in which you can see the interaction of the genes. Here is an example of plants, a small part in which you can see the genetic variation that involves quantitative loci. So you see here a small number of genes or QTLs that have a large effect. Some they have moderate effects, and very large numbers have minor effects. And then you can see here in this heat map, the heat map shows actually a block of genes that interact in one way or another. This is a beautiful example of the very complex epistatic interactions of different genes. We have this information only for model species. I talked about yeast, Arabidopsis and a few other higher crops for which a lot of research has been done. A lot of statistics, which you see here, that you can fine-tune the information that leads you to how genes are interacting with each other. So we see that apart from the epistatic effects, you also have the effect of the heritability, the environment. So the variation in the phenotype. And the geneticists, we use here the heritability, which is a measure for the variation of the phenotype. In other words, heritability is a statistic that estimates how much of the variation in a phenotype trait in a population can be explained to genetic variation. So you want to know how much is genetics, how much is the rest. So consider here multifactorial polygenic trait. You have a component that is the genetic variance here, you have an environmental variance here, and that is too complicated but important to keep in mind. The genetic variation can be an additive effect of recessive alleles, as I showed briefly in the previous slides, can be dominant alleles, very few, and you have a lot of epistatic interactions. Now, what everybody does who works with QTL mapping in crop species, we always want to calculate the heritability because we want to know how much of our observed variation is due to the genetics. For that, we use this formula. This is a so-called broad sense heritability. So we look first for the variance of the environment. So you take this variance plus that variance plus that variance, you average them, and you get 2.28 days is the average of the three variances. The broad sense heritability compares the variance of the genotype compared by the general or by the variance of the genotype plus the variance of the environment. In this case, you have 11.89 is calculated here, divided by the total is 14.26. And you see a value of 0.84. That means that the broadband heritability has a strong genetical component. It is always between 0 and 1. Heritability does not indicate the degree to which a trait is genetic. Rather, it measures the proportion of the phenotype variance that is the result of genetic factors. But this broad sense heritability is not always useful for many cases. So you see narrow sense heritability, and if you look in detail, you will see that the statisticians and geneticists created several different approaches. Here is the narrow sense heritability, in which also the component of additive genetic variance, dominance variance, and epistatic variance are included in the total calculation for heritability. And Ajahn Pumipat showed me that the ANOVA model can be used to calculate also the narrow sense heritability. So that is, and if you look in R, there are several programs that include an heritability script that you can use to calculate the heritability. Now, the quantitative trait loci, we now come to that in detail. What are the properties? It is a region on the linkage map that correlates with the variation of phenotype. Such phenotypes vary in a degree and can be attributed to polygenic effects. That means you have the product of two or more genes and also the environment. And the QTLs, unlike continuous trait, those traits can vary continuously. Think about flowering time, height, weight, length, and that sort of things. A single phenotype trait is usually determined by many genes, of course. Consequently, many QTLs are associated with a single trait. It can be mapped in experimental populations or in natural populations for respectively QTL mapping and genome-wide association study. And QTL and genome-wide association mapping is now possible thanks to a large number of markers, molecular markers, thanks to genomics information, and certainly also a lot of advanced statistics. The method that I'm going to discuss later is the method are employed to map QTLs, which each with the strengths and weaknesses. Several common used methods is linkage mapping. Simple linkage mapping involves creating genetic maps with polygenic markers like SNPs, microsatellites, segregating in a population, and these typical involve phenotypic assessment. So you have to assess the weight and so forth, the color, many things. Happily enough, nowadays we have automatic or robotized phenotyping methods in which a large number of phenotypic properties can be measured in an automatic way. And that reminds me that in my university, we have Arabidopsis grown in growing chambers. And we have a camera that every second makes a photo among the few hundred drills that are growing there. But not only in the morning, also at noon and also in the afternoon. And to my great surprise, in these genotype and phenotype assessment, they told me that the position of the cotyledons in the morning are low, in the middle they are widespread, and in the afternoon they are high. And if you do a QTL analysis, you find QTLs for the different positions of the leaves in this, that, and that way. So you see that the phenotypic assessments, the more accurate you can do, and the more controlled information, the more information, the more QTLs can be detected in your material. And then the statistical analysis I'm going to talk about in a while. Now, very simple. How can we find a quantitative trend? If you take Arabidopsis as an example, one is early flowering, the other is the late flowering. For rice, you can do the same, and so forth. You compare, you build, you put together all the plants that are early flowering and all the plants that are very late flowering, and you compare every chromosome for the red and the blue segments, and then you see, what to your surprise, of course, that all the early flowering have here a segment which is blue, and the late flowering has chromosomes, same chromosomes, in which you have the red markers in that region. So that visualizes very clearly that in this bulk comparison of early flowering and late flowering plants, you already have an impression where your QTL is located. So the early flowering is a quantitative Arabidopsis. The bars represent one of the 5 Lingus groups. Red are part of the Lingus group marked from parent 1, blue from parent 2. And in the second population, you compare the plants that are early and those that are late flowering. Now, I was struggling, to be honest, when I was a teacher, I was struggling a lot how to get the QTL phenomenon, the concept clearly. And my colleague gave me an Excel file, a simple Excel file, and you can use that and you understand. So it is an Excel file, a very simple approach in which we compare the flowering time, as I showed you in the previous slide. So here are 23 plants. Actually, we used 106 plants and many more. And we have 6 or 7 different markers. Marker 1, marker 2, and for every marker, we score the quantitative trace, so the days of flowering. How long does it take for the seed to develop, finally to come with the first flower? That number of days is here. And for every marker, we have the A allele or the B allele. Now you get these kind of Excel sheets, and you can imagine that in the final, when you do it on a large scale, you have many more data. But the idea is the same if you compare to a very simple statistic. So here is marker 1, and you see here the scatter plot for the alleles A and B, and you see the average, the variance between A and B is almost the same. But for marker B, you see a very clear difference between the A and the B markers for the second marker. So the simple method is marker regression. Calculate a t-statistic, or you do an ANOVA to compare the 2 marker phenotype. And then you see clearly this difference is not significant, and that difference is significant. So we have here, the conclusion is we were able to say, okay, my early flowering is located somewhere on a Lingus group, but also M1 is located close to that. So, ladies and gentlemen, I hope you can still follow. I'm afraid that now we get a bit more complicated with the QTL and GWAS programs. But now, in my previous hour, I explained to you that with a simple QTL, comparing all the plants that have the QTL or don't have the QTL, it's quite straightforward. But the solution to that mapping is quite simple. because I already mentioned that the QTL can be influenced by many other QTLs. So you need more advanced QTL mapping strategies. And one is the simple interval mapping, which actually is a continuation of the example that I use for my students to map a simple QTL somewhere on the chromosome. So here you see a simple lot curve of the simple interval mapping of chromosome 2 of maize for a cell maize in concentration. Okay, so the single QTL assumption generally works on the idea, the assumption that single QTL influencing the trait with the interval defined by two flanking markers. So you have two flanking markers and you know, okay, my QTL is clearly associated with one marker, but also associated with the other marker. And you already did a genetical mapping, so you know that the two markers are somewhere on the chromosome, so your QTL must be somewhere between these two markers. What you need is a dense map of markers to pinpoint the QTL locations effectively. That is nowadays not that easy and not that difficult if you have, let's say, physical mapping, or you have markers, a lot of markers, let's say 100 markers per chromosome. That already is a good starting point to find your QTL markers. Now, this method is very straightforward, but you also know it has a lot of drawbacks and limitations, especially when you have multiple QTLs that influence the trait, leading to an inaccurate localization. So, what I mentioned here, you have here a couple of markers with a clear lot support showing a lot of 2, in which there is an effect of the QTL here in this region, and the peak of this profile suggests you that your QTL is somewhere between these two markers. So you can calculate the log profile like here after interval mapping with a maximum likelihood or a regression, two methods that are quite often used in statistics. And the same what I mentioned for the lot calculations in linkage mapping. If you have a value of 3 or more, you know that it is most likely that you have QTL under this linkage. But still, you can always argue, if you have more of these hills on what they call hills on the landscape, where is your threshold? Where do you put this line, right? And in QTL mapping, people use a lot the so-called permutation test. The permutation test actually shuffles your phenotype data, but keep the markers constant. And you repeat that work 1,000 times, and then you determine what is a significant level that such false positives is below a certain level. So the permutation, I will come to that quite more extensively. The permutation test is a very good and reliable method to find what is above permutation. It's interesting for you to map your QTL, and what below is we call it background or noise. What's that? But as I also mentioned, keep in mind that you have multiple QTLs that play a role in the same experiment. So we need a multiple QTL model mapping. And what is it? It is a composite interval mapping. It's more or less the same. It is in the computation, in mathematical computation, it is slightly different. But I see that in the professional QTL mapping that I look for, they use actually both MQM and CIM. There's both multiple QTL models and composite interval mapping to find your QTLs. And in the good case, you find probably the best, the same QTLs. So, to make things a little bit clear, if you have a QTL mapping profile like here, you see, okay, there is a top here, but there might also be under this position, there might be a second one, And that I did it in a simple Excel calculation. You can assume that you have two QTLs producing the same quantitative rate, and you put them together in the same observation. You see that if you calculate these two together, you get a profile like that. So QTLs at other positions introduce variation in the trait values for genotype classes at the test position. So when you test for a QTL at a given position, the effect of other QTLs needs to be eliminated. So when you get a better position on what we call the major QTL, the most important QTL in your data set, you have to remove the effect of this guy. And I think that is very beautifully demonstrated in this interval mapping simulation. So you see here the 5 chromosomes of Arabidopsis with the lot score. There is already a permutation and threshold, and this region here, that region, and that region are potentially interesting places for the QTLs. Well, that's quite a lot, a bit too much to make straight conclusions. So you want to eliminate the effects of the minor QTLs, right? Now, what are the minor QTLs? Minor QTLs are cofactors that are also have an effect on the QTL, but these effects can be beyond the threshold level, beyond here this dotted line that you see in the middle. So you can calculate the cofactors that you assume here in this method. You can calculate them with the program and say, okay, these are my cofactors. Use the cofactors to eliminate the effect on the major QTL. Or you can, in the case of MapQTL, I don't know for RQTL, but in MapQTL, there is a tool who can find you the cofactors and see what the effect is. So if you take these cofactors, these positions, the cofactors, then you see that the QTL patterns completely dramatically is going to differ. It's going to be so you get very steep positions here and there, and you get a lot more effect of the QTLs when the cofactors are considered, when the cofactors are actually used to sharpen the position of the QTLs. The cofactors are other genes that epistatically interact with the main gene. Now let us see. I mentioned so far the very simple interval mapping, the multiple QTL model mapping, which nowadays I believe is used in most programs, and the composite interval mapping. So for the QTL assumption, you assume that you have a single QTL per interval between two or more markers, but in the multiple QTL, you have multiple QTLs, and also in the SID, you also assume that you have multiple QTLs. The method here is a likelihood ratio statistics, what is about a certain level that is considered. In the MQM, the regression modeling, and in the SIM, a likelihood ratio with background adjustment. So in this case of interval mapping, you have no background correction. In the MQM and the CRM, you do have a background, as I mentioned, as I showed you in the previous slide. This is simple, and these two are more complex, especially the CRM is complex and arithmetic. But the marker density in all cases should be high to find the reliable position of the QTLs. Okay, now I have given here a summary of the comparison between MQM model and the CRM. A lot of background information. You can have a look at it later on when you are at home. I understood that you can download my presentation, which is almost the same that is available for you. There are a couple of points that I want to mention to you. The pros and the cons in QTL mapping. A couple of things that you have to keep in mind. First is that the complexity. If your complexity higher, then you see on this place, by fine-tuning, with a higher resolution, the position of this QTL here on this chromosome can be located on the very end of this chromosome. So you see by the more accurate higher marker density, you see beautifully here that this position is still a lot narrower than considering QTL in the whole chromosome. This still, I have to say, is too high to find the genes. You have to think about recombinant nils or other methods to narrow down very much the region where your QTL is located. Let's say if this is a tomato chromosome, then tomato chromosome can be, 800, this is 100 K, then you have here a region of 10 to 20 K base pair that is still far too big to find the putative gene. So you have to do additional experiments to find your point of interest. There's another thing, what happens if you see on chromosome 4 that there is no QTL, at least you have some effect, but if you use the permutated threshold, it is below the threshold, so you don't consider. But when you enhance the complexity, then you see on that chromosome, you see a positive QTL on the end, a negative QTL is a negative effect on the quantitative traits further on, and also a third one. Now these two, and then together, creates a situation that at low complexity, at low resolution, you do not find the QTL, whereas there are 3 QTLs in the peak. So you need to do a QTL mapping at high complexity and resolution. High complexity means more time, resolution also needs more markers. Another thing I already mentioned that to you, that is epistatic interaction. Of course, if you look at many quantitative traits, several QTLs may be involved. And here, if you have 2 QTLs, if you have QTL chromosome L under the L for chromosome 4, or you have the L chromosome with the K allele chromosome 4, you can see a dramatic effect. If you look for the K of chromosome 3 and the L chromosome pair, compared to the Ks of chromosome 3 and 4, Ks and Ls are the names of the alleles, then you see there's a dramatic difference in the effects of the QTLs, showing a clear epistatic interaction within these 2 QTLs. You also can see it here in the profile. Here is a lot more difference between them. So epistatic interaction is important, but the more you are going to do the analysis, the more markers you have, and the more analysis, you see that the effect of the accession layer and cont can be separated, clearly showing an epistatic interaction between those QTLs. There's another one. That is a picture that I got from a Nature Genetics review paper. Very clearly, in the left you see the effect of 3 genotypes, phenotypes, with an additive gene action on locus X, partial dominance at locus Y, and no epistasis. So you see that they simply have quantitated more effect, but it is not different. They simply are additive. They sum up the effect. In the middle, you have clearly an cases of epistasis, in which this one has a negative effect compared to the red and the green line. So indeed, the additive effect of locus Y is much greater in the X1 genetic background than in the X2 genetic background. And in the right, you have very complex situation of epistasis in which the additive effects of X are close to Y1, and Y2 genetic background. So you see here a situation which is very realistic, in which one QTL clearly genetically influences the effect of another QTL. There's another thing, I do not go very much in detail, but you have to know it, and in the literature, when you work with model species, it is interesting to show that you can focus on the QTL mapping based on regulatory elements. And why it is important? you can imagine quantitative effects are based on a regulation on the amount of gene expression. This expression quantitative trait loci. If you look at the phenotype, it will not help you much. But what you can do, you can look at the expression levels of microRNAs, messenger RNA, not microRNA, messenger RNA. And messenger RNAs will clearly show you an effect between the eQTLs of the different markers. So the measure of the microRNAs or protein traits are almost always the product of a single gene with a specific chromosomal location. So you have less effects of others. You can very much pinpoint to the expression of a single gene. Cis eQTLs on the side of the gene of origin, trans when it is far from the gene of origin or far away. And some cis are detected in the same tissue, but the majority of trans eQTL are tissue dependent. So that is one of the things. When you isolate messenger RNA, make sure that you always take the messenger RNA one and the same tissue. So after isolation, the messenger RNAs, you do always an RNA-seq data as a micro should be messenger RNAs. So we have seen that QTL mapping is robust. It can help you a lot, especially when you have a high density map, but still the resolution is low. We're now going to the second map, and it is the third part of my presentation for us to do, genome-wide association mapping. I mentioned that for the QTL mapping algorithms, we can use F2 or we can use the RILs and the NILs, so in which we compare different genotypes with the effects and look for the effect of QTL. Not surprisingly, you will see it is a powerful method. It is not very accurate. Whereas in the GWAS, you can calculate, you can work with a panel of plants, and also plants here that contains genetical different parts that obtained from original accessions or wild relatives or cultivars. You can make a panel of by many generations that contain a large genetical variation. And we're now coming to the association mapping. It is a statistical method that identifies QTL by examining marked trait association that can attribute to the strength of linkage discipline. Don't worry, I'm going to explain to you what that is. Between markers and functional polymorphisms according to a large panel of different germplasms. Linkage disequilibrium is related but not the same as linkage, but it is in the population genetics term that we use for a non-random association between two or more loci in. In general, the closer two markers are, the stronger the LD, the stronger the linkage disequilibrium. And you see these magic words in general, that means there are exceptions. Not a linkage mapping, but in LD, there are. And depending on the marker coverage, the candidate gene association study can you use in the prior information. So if you already know somewhere where the gene is located, let's say you have already obtained information by QTL mapping, so you can focus very much on the precise location of that marker in genome-wide association study. I made here an overview in which you can see what are the differences between the traditional QTL mapping and the association mapping. I already mentioned to you that the populations that we use for QTL mapping can be F2 or VC1s or RILs, NILs, and the like, right? So that you use for linkage mapping. But for association mapping, you don't need to make this kind of things. then you have to make a panel of plants that are different for a large number of markers, for a large number of genotypes. In the genotyping, for the traditional QTL mapping, you use a linkage map. Linkage map used to make QTLs. The number of markers, at least 100 to 400 for framework map. But I mentioned 100 for a chromosome is well the least that you can use. For the association mapping, you're not looking primarily to a linkage map, you look at a high marker density. The number of markers in general, don't worry, it's more than 10,000, aligned to the reference physical map position. The phenotyping is actually the same for the T phenotyping field and greenhouse and lab-based files, but in the association mapping, you can do a lot more by appropriate applications and experimental science. That is where the statistician comes in and will give you a lot of suggestions. And then for the analysis, I meant to you have interval mapping, different interval mappings, and for the association, we have association mapping based on structure analysis, kinship, and links discipline. That is what I already mentioned and will explain more in detail. For the significance, to see what is relevant, I mentioned already the permutation test, which is very powerful for the association mapping, where you have a lot more information. You have data sheets that are much, much bigger. People use the Bonferroni correction, false discovery rate, which is in generally more recommended, and even permutation tests if you have a very powerful computer, because the calculation takes a long time. It's very, very arithmetic intensive. So I'm going to mention a couple of advantages and probably in your version I converted opportunity and challenges. I think it makes sense first to stand still with the opportunities of GWAS. It is a multidisciplinary approach in which you use a lot of statistical computer power and a lot of information, sequence information, and it can unravel physiological processes. But the challenge is you need a large population size. You need increased allelic variation. That's why you make these panels with a lot of different varieties or cultivars. So you need to identify the extreme accessions, why that is important both down and wild, and you improve the accuracy trait correlation. And you need high density maps. Maps only if you make the correlation with the QTL mapping, increase resolution, and increase the efficiency in genetics. The pitfall, and that is what we're going to discuss now in a while, there are severe things you have to take care in your mind, that you have multiple QTL alleles, the same as I mentioned to you in the QTL mapping. You can have rare alleles, and you can have many small effect QTLs. Do you see them? Do you not see them? That is a major question. And the population, this structure, is a major point of concern in new GWAS. And then you have, what you always see people talk about, false positive and false negative errors. What are people talking about? Now here you see in a nice example of what people call the Manhattan plot. Manhattan plot is blue, right? You can see, okay, these are my 7 barley chromosomes, no problem. And I have no significant signals for chromosome 1. For chromosome 2, okay, you have here a clear point. In 3, I don't have. In 5, in 4, I have. 5 not, and so forth. But this may be clear, that may be clear. But there are still some, like this one, are they serious or not? So you have always something in your mind that you have a type 1 error. That means that you can ask a person like me, and the doctor say you are pregnant, then it is quite likely that the doctor makes a mistake, right? It is false negative. And if this young lady comes to the doctor and the doctor say, you are not pregnant, now then you have probably severe problem, but most likely this doctor has a type 2 error, false positive. True positive, and a false. So you see here that the false positive and false negative errors, they are very important to judge later if these peaks, but also these peaks and these peaks, do they represent true QTLs in your analysis? That is why in the GWAS analysis you have a large number of statistical tools that I'm going to explain to you just briefly to give you an idea what is important. The genome-wide threshold, the significance threshold, as the permutation that I mentioned before. Linkage decibelium that is going to be tested, the market trade correlation, the mixed models, I will mention that very briefly, the population structure, and the PCR, the principal component analysis. Kinship testing, which goes hand in hand with the population structure. And finally, I mentioned QQ plotting. In the end, you see in papers that are now downloaded and read about GWAS analysis in plants, quite often you see that they present population structure kinship and QQ plotting, and they use mixed models to improve the final result and interpretation. Okay, now let us look for the significance. I mentioned already to you that in genome-wide association, a lot of people use the Bonferroni correction. You divide your alpha level, which is mostly 0.05, by the number of tests that you do. While it is simple to implement, it can in large dataset lead to more type 2 errors, failing to detect true positives. False discovery rate is the test that you use to control the expected proportion of false positives among your declared significant research. It is less conservative than Bonferroni and allows you more discoveries while still controlling false positives. False discovery rate is handling the genome-wide association, focusing on balance, sensitivity, and specificity. And then what I will not discuss in detail because it's very complicated, the people use more and more in these statistical approaches Bayesian statistics, and it is also for the false efficient false discovery rate is a Bayesian alternative. I don't know how many of you got Bayesian statistics in statistical class. I see some of you starting to laugh. It's a bit complicated, but statisticians, they really love it more and more. It's getting more and more in a wild field of computations. Anyway, this is a new way of testing the significance. I mentioned permutation test. That is very calculation intensive, but I think it's more reliable than the Bonferroni. And very recently, I found also the simple M test, which estimates the effective number of independent markers. Now, this will help you a bit more. So if you have a data set, for the data set, you do a Bonferroni, the simple M and the FVR, and here it is a matter of statistical feeling and emotion. You authors say that the simple M is the best thresholding in your material. I don't have the expertise to say if they are true or not. Bonferroni in general says it's conservative and not. You may miss some of the false negatives. But anyway, you see there are significant thresholds that you can use to see what are your important data. Then linkage decibelium, I mentioned to you, it is the non-random co-segregation of alleles of 2 or more loci. It is different between observed and expected allele frequency, and especially assuming random distribution due to independent disorder. This non-random co-segregation in general could be between loci on the same chromosome, can even be on different chromosomes. So I already mentioned to you that linkage discipline to a greater part can be compared to linkage in a linkage mapping, but in some cases, if you select for other markers on other chromosomes, then you can also clear linkage disequilibrium. After many meiotic events, only loci that are tightly linked to the functional mutation will show significant association with the trait of interest. In other words, linkage will give you an information about the meiotic history, the generation history of your material. Linkage means also that the distance between the marker and the seemingly tightly linked QTL may be far greater than thought, making method-based cloning of the gene to develop the best markers highly improbable. It may also result in miscalculation of the QTL effects. And process that can affect the linkage disequilibrium is in epistasis, gene-gene interaction, natural selection or breeding, important for the breeding work, mutation, random shift, genetic hitchhike, and gene flow. And I mentioned why is LD testing so important. Now LD defines you how far you are allowed to look from the detected markers when to look for the cause, for the causality on the genome. So the which of the QTLs are really located or very close to the markers that we have in our panels. The LD shows the association between SNPs and an unobserved gene or QTL. The degree of LD can be different between species and between generations. And that can be a difficult situation in your analysis. The power and resolution depend on the extent of LD in your map quantification. And LD can help you to the so-called synthetic association between the genes that are causing, positive, or sometimes away from the GWAS mix in the manhattan. And the LDDK pattern, which I'm going to show in a while, is affected by the recombination rate and the number of generations recombination. Therefore, study of the links disability decay may reveal the population recombination history. And for the LDDK, it helps to estimate the number of markers that are needed. If you have a very strong linkage disability in a certain region around your QTL, you may need many more markers in that region. Now I found, and this is my only picture which is not plant related, it's a very nice example between linkage disequilibrium and the structure, what we call the LD block structure, between two strains of the chicken. One is with the brown and one with the white. They are completely different strains. And if you compare on this LD block structure, you see a very different LD blocks resulting from a long-term breeding history between the brown and the white chicken. So now we have to realize that such differences that can result from population structure, there is an allelic variation between subpopulations that will become later in a while. Genetic drift, that means you have a random fluctuation in small populations, not applicable in this example. Natural selection or breeding, that is very much applicable in this situation. And the recombination rate and the gene flow, non-random mating and bottlenecks and environmental effect. But I think here that especially the natural selection or the breeding of these two strains of chicken. And here you see an example of linkage disequilibrium decay plotting. This example shows you all the markers and the map distance. So from here, QTL to let's say 40 LD. So this is independent. and this is highly linked. And you can see this red line here is very sharp decay showing that your strict linkage is here. Now, you can imagine that when you do this for a genome association panel, that you get a fairly sharp decay, but if you use a real population like here, that your decay is much less sharp, and then it is more difficult to use. But one of the problems that you can have, if you have a linkage, you have here a situation of two linkage groups on the same chromosome, and it's a recombination hotspot here in between, and physically they are close together, but in the end you have two linkage disequilibrium blocks that are apart. So you see a pairwise linkage disequilibrium that is plotted against the genetic distance, and people use always Pearson correlation coefficient, the R squared. The inner fitted line is a non-linear regression curve of these R squared on the genetic distance. I mentioned that the LD decay is considered below 0.1 threshold and based on trend line, and this around 30 to 40 centimorph. It's about this one. A pairwise linkage disequilibrium between unlinked marker loci is assigned to 100 centimorph distance. Now here you can see a very nice example of three Manhattan plots with the marker here in the middle, QTL, and you can see here that in this region you have linkage disequilibrium block. The light arrows here, the light regions, is a lot of recombination. There is the LD is low, and the dark red are the regions where the LD is high. And here in this region, you have much less recombination, and the LD is much higher. And in this region can be in a region full with repetitive sequences or in heterochromatin. And you see here that in this region, the LD block is very high. And it is difficult to find in this region where your gene is located. In addition to your QTL analysis, people use market trait correlations. So you see here in model species for which you already have a large number of genes, you can see in these genes that you're mapping the chromosomes. What is their correlation? And here in this case, you have a couple of related genes that are highly correlated. Here is also a region with a highly correlated block that gives you clear correlation, how different traits are correlated. You can use it to identify genetic association and understand the basis of the traits. This plays an important role in the selection for breeding, and it can be used to improve the trait prediction and exploring the environmental interaction. Okay, so I mentioned already that population structure is important. And two statistical phenomena are used for the relatedness among genotypes. One is the population structure, which is essentially the systematic difference in allele frequency between subpopulations caused by geographical and climate change family relationship. And this can lead to quite a lot of false positives. And the second is the so-called kinship, the unequal familiar relationship. It's a familiar relationship from recent ancestry among individuals usually exists in different panels, which can lead to wrong, spurious associations. And estimate relation among individuals using molecular markers due to incomplete pedigree information, the kinship matrix, which are shown on one. Two individuals sharing a lot of genotypes and SNPs are likely to belong to the same family. So first about the population genetics, I mentioned it is a non-random structure of alleles at different loci in a given population. Loci in linkage discipline when the frequency of association of the different lines deviate from being independent and associated randomly. And so patterns of linkage discipline in a genome is a powerful signal of a population genetic process that are structured in it. And this requires additional statistical analysis of a population analysis. Now, this is with program structure of fast structure and also a program like Emma, which I did not see. You can see how far you can put threshold. This is rise. In a rise, you have with a so-called K value. The K value is a hypothetical number of populations, and you look for the optimal number of subpopulations. Now here you have the group of the Japonica rise and the group of the Indica tribe. But if you make the number of subpopulations more strict, you can see here, there is also an Australian group of rice that's different. It is Indica, but it is distinguishable, and the more precise you go, the more differences you have. Now, what the K value, which is very important to calculate, is an approaching or a plateau that continues increasing. And here you can see in this one, if you have K1, all the rice. K2, you have the distinction between Japonica and Indica. Here is also the third one, Australia, and so forth. So if you increase the number of Ks, you see that the number of subpopulation increases. But that is not workable. You won't actually go to about 6 or 7, because here the plateau is, the increase is very, very horizontal. You don't see many more subpopulations. For a true value of K, you find the smallest value of K that captures the major structure of the data. So once you have seen that there is a population structure, in the statistical approaches, you have looked at the linkages implement to correct for it, but there are also different methods to correct for the errors that occur in the population structures. And one of the methods to further analyze the population structure is a population, the PCR, principal component analysis. Here you have a principal component analysis in which you have the principal component 1 and 2, which you compare. Now, most of these plants are beautifully on a straight diagonal, but if you use the principal component 2, loading factor 2 and 3, then you can see that you can separate the subpopulations, giving clear information about the relation of these, the population relation of these different plants. So the principal component analysis is a technique to emphasize the variation and bring out a strong pattern in the data. It is used often to reduce the number of variables and so to make data easy to explore and visualize. So the principal components are a linear combination of original variables. So the PC2 is a recalculation of the PC1, in the PC3 also the PC1 and 2. So the first principal component is the direction along which the samples show the largest variation, in which you can clearly cluster them, you can separate and reveal the nature of population structure. Here also in the kinship analysis, you can see here in GWAS, it refers to the relation or genetic relatedness between individuals. So here you see large number of individuals, and you see this block is a block of individuals which are highly related also. This block as a whole is related, but within this block you see that these are very related. The K matrix estimates the fraction of the shared alleles. So this is all based on shared alleles or shared fragments of haplotypes in the population. Now, if you have all these problems, you will notice, and tomorrow or the day after tomorrow, it will be explained by Ajahn Bhumipad, that you use mixed linear models to account not always for the fixed effects like the SNPs, but also the QTL model, the population structure, the kinship, and the errors. So the variation that you obtain from the population and the kinship analysis can give you variables that you can use here in this mixed linear model framework. Don't worry, there are many more than these. I mentioned here the general linear model, there are the mixed linear model, but there are also several others that can be used to solve the problem of the population structure. So in this unified mixed model framework for association mapping, you can see the multiple levels of relatedness. And by using these kind of mixed models, you can control an equal relationship using the population structure and the kinship matrix. And that brings me finally to the QTL plotting. I found a very nice explanation on YouTube, which for those who like it can share with you. So it complains that the quantile are actually the expression of the genes, the expression of the genes, shown here as these dots, and you can compare them with the normal distribution, or you can compare them with the uniform distribution. So the QQ tells you actually how random your markers are separated and how in GWAS you use them to plot quantile distribution to observe p-values of the y-axis versus the quantile distribution of the expected p-values. To compare this later with the Manhattan makes a lot clear how QQ plotting is used in practice. So I mentioned already the mixed models, and the mixed models is a statistical method that dramatically reduces the inflation of the values. So normally in the ideal situation, you have the gray line with a few exceptions, and these can be the points that you find above the threshold in your Manhattan plot. But if you have a very high, a very definite quantile QQ plot, you need the mixed model to dramatically reduce the inflation of the p-values. Accounting for the population structure with the mixed model dramatically reduces also the inflation, as that you can see also. So this is uncorrected, and this is the corrected one that clearly support your identification of your significant markers in the genome-wide association. Now here you see a few examples. This is in the first example, GWAS resulting from an Anderson-Darling test, and to capture a very definite compute prompt that you really can see, oh, your data are not reliable to see what are the false positives and the false negatives. But if you use the GWAS resulting from a mixed linear model, and you can see, oh, now my points are really very close to the theoretical, and now we can clearly identify this QTL on the Manhattan point. That's your possible signal. So you can see here that the correction of your with the mixed linear model can help you and show you in the QTL what is the reliable information. I give you just a few more examples how the mixed linear model can make corrections. So you see here examples of the model of the population structure and the kinship, and you see when you correct for both, then these QQ plots are very close to the theoretical value, the diagonal that you mentioned. And also this is a paper in which people use the simple and the third of method to model to correct for problems in the genome-wide association. Now with this last picture, I just show you that if you do a GWAS analysis, you need really a bunch of different statistics. First, to show the genome-wide association. Second, to use the kinship, the QTL pattern, with PCR, and so forth. And you correctly mix the linear models to get reliable information about what in your Manhattan plot are the reliable markers. And with these words,